вами сегодня выступит бывший директор программы «Геном человека», доктор Чарльз Кантор. Доктор Чарльз Кантор также является полным профессором университета Бостона и полным профессором университета Калифорнии. Также он является сооснователем компании Сигеном, компания, которая является лидером в неинвазивной перинатальной диагностике и неинвазивной диагностике рака. Также один из лидеров на рынке масс-спектрометрии для генетического анализа. Он также основал множество других компаний, включая компанию Ретардоп, которая использует изотопно укрепленные органические соединения для борьбы с различными возрастозависимыми заболеваниями и старением, и многие другие компании. Приветствуем доктора Кантора. Доктор Кантор.
You want to try to, okay, so good afternoon. What I'm going to talk about is trying to use DNA obtained from patients to make medically useful decisions about patient care. And what that should evoke for most people is measuring a person's genome sequence and then trying to make predictions based on that sequence. And that's not actually what I'm going to talk about. I'm not going to talk about that today. Because honestly, that doesn't work very well, as I'll try to show you before. Because when we measure a person's genome sequence, we're measuring germline. And it turns out that germline genome, for simple genome, has very useful predictive value. But for complex traits, it doesn't. I'm really going to be talking about using somatic DNA samples that may be different in different tissues to make clinically useful decisions. And you'll see that we've made a lot of progress in the past few years in being able to take DNA non-invasively from a patient, mostly from blood, sometimes from urine, and draw inferences about those patients that today are being used in the clinic. So an outline of the talk is I'll show you some of the thinking about germline DNA to try to convince you why it doesn't work. And then I'll show you examples of using somatic DNA in two applications: prenatal diagnostics and cancer diagnostics. Okay, so we know already the genome sequence of lots of people, and more and more people are being sequenced every year. And so it's fair to ask, in the last 10 years, since we've had human genome sequence, has it made a major impact on medicine? And by medicine, I mean not medical research, but actual clinical practice of medicine. And the answer, by and large, is no. Hasn't changed anything. Hasn't been used. There are exceptions. In several areas, in prenatal testing, in cancer, in certain eye diseases. But those are the exceptions. And in most, for most major diseases, knowing a person's genome sequence just doesn't particularly help. Now, the cost of DNA sequencing is getting cheaper and cheaper, and the accessibility of DNA sequencing is getting widespread. So, what I'm trying to tell you is there's a mismatch between our ability to generate this information and our ability to use this information. And here's an example the first genome-wide test that became widely available was a test from a company in the US called 23andMe, which doesn't measure the whole genome sequence, but samples the genome at tens of thousands or hundreds of thousands of sites. Okay? And the first person I know that was analyzed by 23andMe is Mark Chi, who was one of the senior scientists at Illumina, one of the sequencing companies. And the only thing he learned from this test was that he was Asian. Of course, he knew that. And when I speak to other people I know who have been tested, even as recently as a few months ago, it hasn't changed. Most of this information is not used. Now, in the field of DNA sequencing, we're progressing from relatively simple tests like 23andMe to whole exome sequencing, all the genes, to whole genome sequencing, and the 6 billion base pairs of DNA. And so it's fair to ask, as we get more sequence information, is the situation going to improve? And I predict, by and large, that it will not improve. If you're interested in your genealogy, you'll learn a lot, no question about it. But if you're interested in your risk of common disease, as I'll try to show you, you're not going to learn it. Now, if you're interested in whether or not you have a rare disease, and you know your genome sequence, it's tiny. Okay? Rare diseases, single gene diseases, what we call Mendelian diseases, are absolutely predictable from the DNA sequence. But if you have one of those rare diseases, you don't need to know your DNA sequence to know that you have one of these diseases, like sickle cell anemia. You know it already, right? So the problem is that it's hard to predict disease susceptibility from genome sequence. And the reason, now we're talking germline DNA sequence, the sequence you inherited from your parents. The reason is that most common diseases are truly complex, and they mix genetic effects that are horrendously complex and environmental effects. Now, humans are genetically very heterogeneous. If I had the DNA sequences of all of you in the room, any two people would have differences, millions of differences in their genome. And you can't solve a problem with a million inputs. So as we look back over the 10 years where we've had genome sequences, only one complex disease has basically been solved. And that's something called age-related macular degeneration, AMD, which is the most common form of blindness in, with the most, and I better not move my hand. Age-related macular degeneration is the most common form of blindness in old people. 10% of the population develops symptoms called dry AMD, and that's harmless. It's fatty deposits under the retina. You can see them in the eye, but it doesn't hurt you. 10% of those people, or 1% of the whole population, the fatty deposits lead to inflammation that produces the growth of blood vessels in the eye, and these blood vessels eventually block the central focus of the eye, and you go irreversibly blind. Okay, so 1% of all people will go blind because of what's called wet AMD. Now it turns out that the genetics of this are relatively simple. Only 5 genes, only 13 genetic variations, 13 polymorphisms, account for most of this disease. There are environmental factors too, smoking, obesity, that are additional risk factors. Okay? But the genetic factors are very simple. So we can now, with a trivial test, predict your risk of progressing. If you have the dry form of AOD, we can predict your risk and the rate actually at which you're likely to progress to the wet form. And this is useful because we have therapy. And the therapy that prevents progression is a monthly injection into the eye of VEGF inhibitors. It's very effective, but it's not pleasant. You don't want to treat people this way until they need it. And so the way that physicians use this genetic test is to decide how often to do more complex screening of the retina. If a patient has a very low genetic risk, then maybe they can see a retinal specialist once every few years. And if they have a very high genetic risk, they better see a retinal specialist every few months. Because once the progression to wet form starts, you can't reverse it. You can stop continued progression, but the drug cannot reverse it. Okay? So this is one example where whole genome analysis by a whole bunch of different laboratories produced results. And from a scientific viewpoint, 4 of those 5 genes are in the complement pathway. So this is a very simple situation. And undoubtedly, we will see better therapy develop now that we understand which pathway is going wrong. But that's the exception. And most of the other common diseases that have been analyzed by looking at germline, by looking at the inherited variations, have yielded 20, 50 genes involved. So it's too complicated to analyze. In addition, we know that for most complex systems, that the environment plays roughly as important a role as genetics. And we know this in human because we have monozygotic twins. We have identical twins. And we also have dizygotic twins, fraternal twins. And monozygotic twins have identical germline DNA sequences. And dizygotic twins have half of them. Okay? And so by comparing phenotypes in monozygotic twins and dizygotic twins, we can dissect the importance of genetics and the environment. And I can give you the answer from hundreds of papers. It's half and half. Almost anything you can think of, roughly it's half and half. Okay? So, what I've tried to show you thus far is that because humans are genetically very variable, we are not rats. Okay? And because our environment is very variable, we don't live in laboratory cages. The problem of making predictions is just not going to happen in the near future. So when you have a problem that's too complex, the effective strategy is to try to simplify it. And the way to do that for complex disease is to divide these diseases up into subcategories. And you can try to do that by using clinical parameters. But in fact, what's working very well is to use molecular parameters. So, for example, you can use gene expression patterns in a whole bunch of people who have arthritis to dissect arthritis into different subtypes, not based on the final pathology, but based on a sort of molecular picture of what seems to be going on. And what happens then is that each disease becomes many diseases. And there's nothing wrong with that, but you can see what you're taking a problem that's impossibly difficult to solve, and you're dividing it into 10 problems, each of which is still very hard to solve. So progress is going to be slow. Now, oops, wait a minute. This just died. That's the germline. That's the DNA sequence that you inherited from your parents. But what I'm going to focus on for the rest of this talk is what happens in somatic tissue. What happens in the blood, or I'm going to focus on the blood, but what happens in tissue in general, okay? And it turns out that the DNA sequence that you inherit from your parents is mostly constant. There are exceptions. But the way that sequence is used and the way it's modified changes. So the most important thing that happens is something called epigenetics, okay? DNA methylation, which is a sophisticated mechanism that's used to distinguish cell types, distinguish tissues, and it's one of the basic mechanisms that determines which genes are expressed and which genes are not. And we know that DNA methylation is dramatically affected by the environment. And how do we know that? We can start with identical twins. At birth, they have identical DNA methylation patterns. As soon as they're born, their methylation patterns start differing. The older they get, the more different their methylation patterns. Okay? So we know that at the DNA level, although the sequence usually doesn't change, the methylation pattern does. And so this is why that identical twins who have the same DNA sequence can be quite different as they get very old because they have different epigenetics. Now, in addition to epigenetics at the level of DNA, RNA expression changes as identical twins get older. And in addition, I need to remind you that only 1% of your DNA is human DNA. The other 99% of your DNA is bacterial DNA or fungal DNA from all the critters that live on top of our skin and inside our body and in every body orifice. Okay? So you're born more or less sterile. But as you grow, you rapidly become a product of your microbial environment. And we understand now that this is very important in distinguishing health from disease. And it is a very useful clinical signature of what's happening to people. Finally, there are very specialized cases in which the DNA of tissues is not the same as the DNA that you're born with. And the two outstanding examples of this are cancer and in the immune system. So in cancer, the genome is no longer the same as the host, because cancer is a disease of genome instability. The genome starts to change, and there are mutants and various chromosomal rearrangements which make cancer cells different from normal cells. And these changes determine the behavior of the cancer. So they're not just metaphenomena, they're the driving force that makes cancer happen. Okay, so that's one example. The other example is in the immune system, where rearrangements at the level of DNA occur in all kinds of lymphocytes, and they determine the immunological behavior of those cells. Okay? There may be other examples, but people haven't found them yet. Okay, so what I've tried to say thus far is that instead of trying to predict genetic risk, which I think is too difficult, it makes more sense to focus on using genetics to try to find early signs of disease, to try to characterize disease progression, to look at the response to drugs, to look at recurrence in diseases like cancer. Okay? That's the general rule. But there is one exception. The one part of human genetics which turns out to be very simple is response to drugs. Because if you think about it, if I'm giving you a drug, I'm making a very tiny perturbation on your environment. So in a way, it's not surprising that if I make a small perturbation, the genetic determinants that reflect what happens with that perturbation might also be simple. And in the vast majority of cases, they are. So there's a whole field called pharmacogenetics, or now pharmacogenomics, which has grown up over the past 2 decades. And it turns out that by looking at just a few hundred single base changes in your sequence, I have a pretty good idea about the way you're going to respond to most drugs. Okay? So this is the exception. The other possible exception is an infectious disease. So we at least know a few cases where very simple germline genetic changes dramatically affect your susceptibility to infectious disease. And the famous example, if you haven't heard it before, is a deletion of a loop of protein in a gene called CCR5, which is the receptor that the HIV uses to get into lymphocytes. And a small fraction of the human population is homozygous for that deletion, and they are immune to HIV. They can't get the disease. Okay? So there may be more cases like that. Drugs, infections, relatively simple, but complex disease is not. Okay, so what we're going to talk about in the rest of this lecture is what's called biomarkers. Trying to use DNA or other molecular signatures that allow us to make clinical decisions in an intelligent way. And, oops, wait a minute. Yes, okay. There are many candidates for biomarkers, but I want you to focus on DNA because I think it's the best. And I think it's the best for 2 reasons. If I can see a DNA change, it's usually causative. It's the root of the problem. And it turns out that my ability to measure in clinical samples DNA far exceeds any other type of molecule for 2 reasons. DNA is very stable, doesn't change. You can even get it from fossils. And I have the polymerase chain reaction. I have a universal way of amplifying any DNA sequence to any extent that I want. So I just have power that does not exist with any other kind of animal. Okay, now, nobody likes to have needles poked in them or pieces of tissue removed. Okay? So it's pretty obvious that the ideal situation to sample a biomarker is to try to do it non-invasively or minimally invasively. And so if you think of the possibilities, you can look in urine, you can look in the mouth, in feces, but blood is the best source. And the reason is purely practical. We already have all over the world an established infrastructure for drawing blood and doing blood-based testing. So all I'm going to propose and demonstrate in the rest of this talk, that we extend, can we do something about this?
Sorry.
Yeah.
One other alternative, let's just try another microphone once again. I'm sorry about that.
Okay, so I'm sounds better. I'm going to propose, can you still hear me? Yes? Can you hear me? Okay. What I'm going to propose is that we take this existing infrastructure for sampling blood and use it to make samples in which we then search for DNA. Okay, now I want you to think about this slide very carefully. In pregnancy, in cancer, in neurodegenerative diseases like Alzheimer's, in cardiovascular disease, in inflammation, in transplant rejection, in many other clinical settings, cells die. Cells have DNA. And when they die, that DNA is very stable. It doesn't just disappear. Now, biology knows this, and it doesn't want DNA scattered about. So most of the time, when cells die, in the process of dying, they chop their DNA up into very small pieces. And that process is called apoptosis, programmed cell death. And the DNA pieces are actually amazingly uniform. They're roughly 150 base pairs in size. So they have 150 letters of genetic code. As far as we know, the chopping is pretty rare. Okay. So in all of these clinical situations, if there's a connection between the cells that die and the blood circulation, these DNA fragments will spill into the blood, where they are rapidly cleared through the kidney. So on average, a fragment stays around for only 15 minutes. But if cells are continuously dying, then new DNA continuously appears in the blood. Okay? So if I look in blood, then I get a continuous picture of cells that are dying. Okay? So if those cells that are dying were to have no different properties from normal blood cells that are dying, I know nothing. But I can learn something. If the cells that die have different DNA sequence or different DNA methylation than the cells in the blood. And in the examples I'm showing you, in pregnancy, since half the fetal DNA is inherited from the father, there are DNA differences. DNA from the fetus is not the same as DNA from the mother. In cancer, it's the same situation because tumor cells have DNA sequence differences from the others. Then if tumor cells die, those sequence differences appear. Okay? So cancer, pregnancy, we should be able to just look at DNA sequence. If a brain cell dies, it has the same sequence as every other cell in the brain, every other cell in the blood, but it's methylation is different. So for some of these situations, like cardiovascular disease and neurological disease, we're going to have to look at methylation. But for other situations, like pregnancy and cancer, we can look at DNA sequence directly.
Excuse me,
Yes?
can I ask a question?
Sure.
When we use PCR to amplify small signals into the blood, can we conserve the methylation?
No. No. PCR erases the methylation pattern by any PCR method known today. So if I want to look at methylation, I have to have fancier tricks. And I have to do it before the PCR, and I will show you one of those tricks in a few minutes.
But you feel very small amount.
It's enough. I'll show you. Okay. Oops, sorry. Okay, now. Okay, so the good news is that in every one of these disease situations, there is DNA that has information about the disease in the blood. The bad news is not very much of it. And let me be a little more specific. If I take whole blood, only one part in 100,000 of the DNA comes from cell death, comes from apoptosis. All the rest of the DNA in the blood is inside the blood cells. And I don't want to see that DNA. So I have to have very, very good methods when I isolate blood to avoid breaking the cells. The reason why this field has been difficult until recently is that all the standard methods for isolating blood broke some cells. So the DNA I wanted to see that reflected cell death was diluted with blood cell DNA that is not it. But we've now got very good methods to isolate blood and not break blood cells. And these methods are so sensitive that if the person who draws the blood is sloppy with the needle and tears a bit of the vein, we see that. We see background from those endothelial cells. So this is very, very good method. But we have to live with the fact that on average, in blood, there's only, in a healthy, normal person, 1,000 molecules of any sequence. And if I look at a pregnant woman or a tumor, of those 1,000 molecules, only 10% may come from the baby or the cancer. So now I'm down to 100 molecules. And if I only have 100 molecules, a problem arises called stochastic, which is a fundamental physics problem. When I have few molecules, I have statistical fluctuations if I try to measure how many molecules I have. And so I can no longer make precise quantitative measurements. And if I have very few molecules, I might accidentally have no molecules. And then I miss an event altogether. Okay? So in this limit, where I have very few molecules, I'm going to run into problems. And I'm going to need fancy solutions to those problems, as you'll see. Now, what makes this problem worse is that if I compare people, the amount of small DNA fragments in their blood, just for normal healthy people, differs tremendously from person to person. And we don't know why. So in pregnant women, there's a 20-fold variation in the amount of DNA in the blood And it has absolutely nothing to do with the gestational age of the fetus. It has nothing to do with the size of the fetus. Just don't know what it is at the moment. And in cancer, it's worse. There's about 100-fold variation in the amount of DNA in the blood. So what I'm telling you is that some pregnant women or some cancer patients have a lot of DNA in their blood. And so the problem of stochastic noise goes away. But other patients have tiny amounts of DNA in their blood. And then the problem with stochastic noise becomes insurmountable. So what we have to do is estimate for each patient how much DNA is in the blood before we start analyzing it. And I'll show you how we do that in a few minutes. Okay, now, there are ways to overcome stochastic noise. And there really are two approaches. If I have a noisy situation, but I can perform replica experiments, then I can reduce the noise by the square root of the number of experiments that I do. So if I do 100 experiments, I can reduce the noise by a factor of 10. But my procedures of looking at DNA in blood require that I start with at least a few cc's of blood. And I normally start with 10 cc's of blood. So I can't do that 100 times and bleed the patient to death, essentially, okay? So that just doesn't work. I can't repeat it. The alternative, which is less obvious, but it's what we actually use in prenatal diagnosis, is to find different DNA sequences which give the same answer. And if I can find many different DNA sequences which have the equivalent information set, I can average not from multiple samples, but I can average within one sample. And once again, I can improve my signal to noise by the square root of the number of particles. And you'll see that we do that. Okay, so this just summarizes non-invasive prenatal diagnosis, and I'll come back to it. This summarizes the correct situation, and I'll come back to that. And I want to just make a comment about what experimental methods I'm going to be using. I'm not going to show you these methods, really at all. But I just want you to know how this is done. If we have a problem which can be solved by a single genetic locus, a single marker, a single base, we use the polymerase chain reaction. It's sensitive, it's additive, it's cheap, it's automated. If I have a problem that needs to be addressed by 10 markers or 100 markers, we tend to use DNA mass spectrometry, which is basically just a fancy way of doing PCR and detecting lots of things at once. And if we have a problem that requires thousands or more markers, then we do high throughput DNA sequencing. Those are the three methods. And what you'll see in the rest of the talk, that for prenatal diagnosis, we're doing sequencing and a little bit of mass spectrometry. And for cancer, we're doing mass spectrometry. But what I want you to focus on in this lecture is not the methods, but the answer. Okay. So non-invasive prenatal testing. We take 10 cc's of peripheral blood from the mother's arm. And as you'll see, we're going to sequence it. We're going to basically do about 15 million sequence reads of short DNA fragments. So we generate a tremendous amount of data. It's not a whole genome work. The way I like to think about it, it's the quick pencil sketch that somebody would do before they meet an auto painter. Okay? So it describes the genome, but in a very rough way. You have to do about 100 times genome sequencing to actually have the whole thing, to have the whole detail. All right. Now, in an average pregnancy, these are real numbers. In an average pregnancy, 13% of the DNA in the mother's blood is from the mother, and 87% is, no, I'm sorry, 13% of the DNA in an average pregnancy is from the fetus, 87% is from the mother. But as I've already mentioned, the range is anywhere from just a few percent to almost none. Now imagine a situation where the mother has too little fetal DNA to detect. If we measure the DNA in that mother, we're going to conclude that the fetus is healthy. Because the mother's healthy. We won't find any sequence abnormalities. And we're going to conclude that the fetus is female. Because the mother's female. Okay? And this is a false negative. And we'd like to try to minimize that. Okay? So, we have to have a way of looking at a pregnant woman, and before we spend a lot of money sequencing, ask, okay, how much fetal DNA does she have? If the fetus is male, this is trivial. Because the mother's female, she doesn't have a Y chromosome. So if we look at sequences from the Y chromosome, they uniquely must come from the fetus. And so for male fetuses, we quantify using the Y chromosome. But half of all fetuses are female. They have no chromosome. And to make things worse, if you're interested, as we are, in looking at what are called aneuploidies, fetuses that don't have the right number of chromosomes, in most cases, the extra chromosome that's going to cause problems comes from the mother. And so there are no sequence differences that can be looked at, right? Okay, so remember I mentioned epigenetics. The mother and the female fetus have very similar DNA sequence, but they have very different methylation. They have very different epigenetics. Oops, I'm sorry. Okay, so what we've done is to search the genome for sequences that have the following properties. In all fetuses, they're methylated. In all mothers, they're not. And furthermore, they fall into the recognition sites of enzymes that can cut those sequences only when there's no method. So as this picture shows, what we can do, starting with a mixture of maternal DNA and fetal DNA, is we can destroy all the maternal DNA. And now we're left only with the fetal DNA. And so we can detect and quantify the fetal DNA. And we do that on every patient before deciding to sequence. And in practice, if there's more than 4% fetal DNA, we go on and sequence. And if there's less, we disqualify the sample. And we have to disqualify about 1% of the sample. Now I'll show you what one of these methylation sites looks like. Let's focus. So this is one place in the genome. And what I'm plotting is the number of methylated molecules in a sample of mother's blood. And if the mother's not pregnant, there are no methylated molecules. Even earlier than 8 weeks of gestation, I can see methylated molecules in a pregnant woman. And after 8 weeks, there's a nice healthy signal. And you can see that it varies a lot from mother to mother. And here's the number of molecules. It's about 100 molecules. So when I mentioned before that I'm dealing with 100 molecules, this is how I know. All right? So this mother, most of these mothers can be analyzed. Okay. Now, we want to look using the mother's blood for properties of the fetus, so that the mother can be either reassured that the fetus is likely to be healthy when born, or to show that there's a problem and then the mother has to deal with that problem. The most common problem in developing fetuses are trisomy, an extra copy of one chromosome. The most famous of these by far is trisomy 21. That's Down syndrome. The other common trisomies are trisomy 13, trisomy 18. They're rarer, but they do occur. As far as we know, all the other trisomies are fatal. They occur, but the fetus dies. As far as we know, all the monosomes missing a whole chromosome are fatal. The fetus dies. Okay? So from a practical viewpoint, if we're looking for defects that involve loss or gain of a whole chromosome, the only ones we actually have to look at are 21, 13, and 18. Oops, sorry. Okay, that's not going to work. Let's go. Oops. Sorry. Okay. Here's a cartoon. And I've colored red the fragments of DNA that come from the fetus, and blue the fragments of DNA that come from the mother. But there are no useful sequence differences here, not at the level of resolution that I've described. So if I sequence this mixture, I don't know the pink fragments, the red fragments from the blue fragments. They all look the same. So what I do is choose random fragments of DNA from the mother's blood, and I sequence 36 bases. And when I compare that sequence to the genome, then if it matches exactly somewhere in the genome, I know which chromosome that fragment came from. So by performing this measurement over and over again, I can eventually make a histogram like this plot, which is the number of fragments that came from each chromosome. And as you can see again in color, some of the fragments came from the fetus, the rest came from the mother, but I really don't know which. If the fetus has an extra copy of, let's say, chromosome 21, then fragments from that chromosome will be overrepresented in the mother's blood. I wish I could tell you it's a big effect, but it's not a big effect. It's a very small effect. So I have to measure this excess abundance very precisely. But I can do that by DNA sequencing because I just make so many reads that I've sampled hundreds of different sequences, thousands of different sequences on each chromosome. So I get around the stochastic noise, and I can achieve amazing experimental precision by just getting lots and lots of data. Okay, so this shows you the results of a clinical trial that looked at 1,800 pregnant women. And what I'm plotting vertically is a statistical measure of whether or not for each mother there's an overrepresentation called disease score, if you're familiar with statistics. And when I'm plotting horizontally, for each mother is the fraction of fetal DNA. And you can see that those mothers who have a high percentage of fetal DNA tend to have a very significant statistical overrepresentation if they're carrying a trisomy fetus. And those mothers who have very little fetal DNA, things begin to change. These results demonstrate that I have pretty good ability to distinguish between the mothers where there's an abnormal amount of chromosome 18 or 13 or 21 from all the normal. So this was a clinical trial. This was published 2 years ago. And what this basically shows, oops, I'm sorry. This basically shows that we make almost no errors. A few errors. Almost perfect test. But this was 2 years ago. We now have improved analytical methods. We have improved experimental methods. And the accuracy of these tests is not perfect, but it's astoundingly good. We make only a few errors per 1,000 patients. And this actually shows data for chromosome 21 on, I think, about 120,000 women. And this was all done in a commercial diagnostic setting. And you can see that with the rarest of exceptions, there's a pretty good distinction between the mothers where things are overrepresented and the mothers where things fall within the normal sort of Gaussian distribution. Okay? Now, where we make a mistake, we can almost always explain. If we have a false negative, if the fetus really does have an extra copy of the chromosome, but we don't see it, it's almost always in a mother who has relatively little fetal DNA. So we have the option of eliminating the false negatives by demanding a higher minimum percent fetal DNA. If we went to 8% fetal DNA instead of 4% fetal DNA as our threshold for analysis, we could probably eliminate all the false negatives. But then we might have 5% of the samples that cannot be analyzed. Okay? So we've chosen to accept a very small false negative rate. And if you're familiar with the invasive tests, like amniocentesis or chorionic villus samplings, these non-invasive tests now are actually better. That's the invasive. These are actually more reliable. But even more interesting than the false negatives is the false positives. Okay? So we see a few cases per 1,000 of where we call a trisomy. So we tell the physician who tells the mother, your fetus has got Down syndrome or one of the others. And the mother decides, I'm going to have the baby anyway, and the baby turns out to be normal. We know why. And it's biology. Oh, I'm sorry. Okay. There's a phenomenon called confined placental mosaicism. The placenta is trisomic, but the fetus is normal. Most of the free DNA in the mother's blood comes from the placenta. So when this rare event occurs, we will always be wrong. The other one, which happens quite a bit, is called a lost twin, or a vanishing twin. Okay? So the pregnancy starts out as a twin pregnancy. One fetus is trisomic. The other fetus is normal. Trisomic fetuses are not very viable. The trisomic fetus dies. But the placenta that it laid down is still trisomic. And so we see the extra chromosome from a fetus that's no longer there. Okay? And we've got thousands of twin samples that have already been analyzed. So this is unfortunately happens, and then there are other things occasionally. All right? But there's another interesting possibility which happens, which I think the first time we saw this, it really shocked everybody. Okay, here's a patient. Z means excess if it's positive, deficit if it's negative. This patient looks like they have trisomy 21, but they have monosomy 13, monosomy 18. The fetus can't be alive. But it is alive. His mother has cancer. What we're actually seeing is free DNA coming not just from the fetus, but also from the tumor. And in this case, the tumor is deficient in chromosome 13 and 18 and has an excess of chromosome 21. That's fine as far as the tumor is concerned. Okay? This is not an isolated event. I'm so sorry. Okay. Here's another case. We saw something similar. It looked like trisomy 18, but monosomy 21, monosomy 13. People in the lab repeated the experiment. You can see how good these measurements are. We got the same thing. Again, this mother has cancer. Okay. So, jumping ahead for a minute, this already shows you that the methods I'm using for prenatal diagnostics can also be used for cancer diagnosis. Okay. But before I go there, I want to give you a glimpse into the future with prenatal diagnosis. So what's commercially available today are the major antibodies. 21, 13, 18, and fetuses that have abnormal numbers of X and Y chromosomes. There are a lot of sex chromosome antibodies. But that's not all we see. This is a real patient. What I'm plotting is the average abundance of DNA fragments that assign along the genome. Here, starting from chromosome 7 and proceeding all the way to the X and Y chromosome. And if you look at this plot, you can see that at the beginning of chromosome 18, there are too many counts. So this patient has an excess of DNA fragments from the short arm of chromosome 18. When we see something like this, we don't know if it's the mother or the fetus or both. We just know there's too much of part of chromosome 18. And we can tell the difference between the mother and the fetus because when we make plasma, we keep the mother's blood cells. So we can go back to the mother's blood cells, the so-called buffy coat, and analyze the mother and tell. Okay. This happens a lot. Okay, here's just a few more real clinical examples. You can see that this is chromosome 1. You can see that at one end and at the other end, there aren't enough counts. Chromosome 2, there are too many counts here. Chromosome 3, you can see there's a region where there's not enough counts and so on. These are called microdeletions or microduplications. Every one of you has this in your genome. Obviously, it hasn't hurt you too bad. But some of them are very bad. Most of them are not. Okay? So some of these have names, pre-shot, many others. Okay? And what you'll see over the coming year or two is that instead of just telling mothers and physicians about aneuploidies. We're going to tell them about these smaller genomic changes, which are clinically understood. And the more we sequence, or the better statistics we develop, the more we see. In fact, so almost 3 years ago, Dennis Lowe and I published the complete genome sequence of an unborn fetus, assembled by sequencing fragments from the mother's blood. Now this was a tremendous amount of sequencing, because we're not sequencing one genome, we're sequencing the mother and the fetus together, and we have to have enough sequence information to tease them apart. So this is not something that can be done easily at the present time. And it can be done. At the moment, it's outrageously expensive to do this. But sequencing costs keep dropping. And in a few years, it'll probably be possible to do this if people want to know the genome sequence of their fetus before it's born. Before we start to do this, people have to think very carefully about how they're going to use that information. Because really, in many respects, you're learning too much at this point. And I think it would be a mistake to start choosing your fetuses based on frivolous concerns like hair color or eye color or whatever. But the information can be measured. So that's prenatal, and that's the long story. And in the time that remains, I'm going to show you the beginnings of the same kind of progress looking at cancer. Okay. Now, I already told you, in both pregnancy and cancer, there's DNA from the foreign body, from the fetus or from the tumor, present in the blood of the host. This summarizes the average amount of that DNA in pregnant women, in healthy volunteers, and in late-stage cancer patients. And I don't think the difference between pregnant women and healthy volunteers that you see on this slide, I don't think that difference is meaningful, because different methods were used. But I think the difference between healthy volunteers and late-stage cancer is very significant, because it's the same method. So patients who have cancer tend to have a lot more DNA in their blood. And it's very tempting to try to conclude that most of that extra DNA is from the cancer. Now, honestly, we don't actually know that today. I think it's very likely, but we just know that there's a lot more DNA in the blood. So here what I'm showing you for a bunch of different late-stage cancers is the amount of DNA in the blood, just total DNA in the blood, in colorectal cancer, breast cancer, melanoma, etc., healthy volunteers. Okay? And you can see two things. Tremendous variation from patient to patient. The same problem we have with pregnant women. So everybody's different. And the other thing that you can see is that some of these patients, once again, have a lot of DNA in their blood. So we're not going to have to worry too much about stochastic noise. But some of them have relatively little DNA in their blood. And so our ability to analyze this is much worse. So when you see data like this, the obvious question is, if I look in the blood to determine the properties of the cancer. Do I get the same answer as if I do an invasive biopsy of the cancer? So cancers are characterized by sequence differences relative to a normal host. And so do I see or do I not see the same sequence differences in the blood of the cancer patient and in the cancer? And the answer don't look at the slide, it's too complicated. The answer is 50-50. Half of what I see in the tumor is the same as what I see in the blood. And half of what I see in the blood is different. And there are two reasons for this. First, stochastic noise. I'm in the domain with many of these patients where I don't have enough molecules to be reliable. And furthermore, my experimental methods are not, in what I've shown you thus far, as sensitive as they need to be. So in this study, which was published last year, they're using methods which are tenfold less sensitive than the methods now there. So some of this discrepancy is probably just experimental, but some of it is real. The second problem is more fundamental, and it's really the interesting one. If I look at a tumor and isolate DNA from the tumor, by and large, I'm looking at tumor cells that are alive. And if I look in the blood, I'm looking at DNA from tumor cells that have died. And this doesn't have to be the same, okay? So there's no reason why I should have perfect accordance, okay? But nevertheless, already, you see that I can draw some inferences. I can non-invasively, not perfectly, but I can non-invasively begin to describe properties of the cancer without touching it by looking in the blood. And I'll show you in a few minutes useful properties. But it turns out that in addition to these sequence differences, these mutations that define the properties of the tumor, the very amount of DNA in the blood is unexpectedly clinically useful. So this shows the survival time of patients over roughly a 2-year period as a function of how much DNA they have in their blood. Patients with very little DNA in their blood have very good survival. Patients with a lot of DNA in their blood have terrible survival. So we've got two kinds of information now. How much is there, and then which sequences are there. Okay. All right, so I'm going to show you one example, which just published. It's non-small cell lung cancer. And many patients with non-small cell lung cancer have mutations in a gene called EGFR. And that defines the properties of these cancers. The patients with this mutation, which by the way is not linked to smoking at all, have improper cell signaling and they have cancer. And those patients can be treated very effectively with several new tyrosine kinase inhibitors. That's the good news. So if you have one of these activating mutations, if you have to have lung cancer, you're better off having lung cancer with one of these activating mutations because we can treat you. But unfortunately, some of the patients who have the activating mutation get a secondary mutation, which is, don't change it, which is called T790M. It's a single amino acid chain. And in these patients, the drugs are not very effective. They're still better than nothing, but they're not very effective, okay? So what was done in this paper was to look at the blood of patients with lung cancer being treated with tyrosine kinase inhibitors using now very sensitive methods, contemporary methods 10 times more sensitive than were available last year, and asked whether they could see these mutations in the blood, and if so, asked whether the response of the patients who had the mutation was, as one would predict, worse than the patients who didn't. And that's shown, Alex, on the next slide. Okay, so just look at the left-hand side. The green patients don't have this T790M mutation in their blood. And you can see that they survive, this progressive progression-free survival, so the tumor is not getting worse. They survive for almost 3 years. But the red patients have this mutation that compromises the drug. And you can see their performance in the clinic is much worse. One example. I'm telling you that although the results are not published yet, there are probably now 10 examples. So this is turning out to be absolutely marvelous medical practice. It's still experimental, it's still just a research protocol. But you monitor the patient while they're getting therapy, and you see mutations come or go that affect the efficacy of the therapy. And in some cases, when you see these mutations, if you have an alternative therapy, you can switch to the alternative. And all this is being done non-invasively. Okay, now, let's go back and let me show you just one last thing. I need high sensitivity if I want to see just a small amount of cancer in a patient. If I have a late-stage cancer patient that has a huge amount of DNA in their blood, and most of it, let's say, comes from the cancer, it's an easy matter. But once I treat that patient, the amount of cancer DNA in the blood plummets to almost nothing. And then my ability to monitor what's happening becomes highly complicated. So what I really need is methods that will, even if there's just 1 or 2 molecules of cancer-specific DNA in the patient's blood, that I can still find them. And what I want to show you is that we have these methods. This is a bit technical, but and I'm going to need the laser pointer back. If I can get this to work. Okay, all right. So here's a situation. Let's suppose I have a 1% of my DNA fragments in the blood are mutant, and 99% are wild type. There's so much wild type that it's very hard to see the mutants. And so I need a trick. And the trick is shown on the right-hand side. It's well, can we go forward one? Alex, go forward one slide.
Actually, you have the flicker.
Okay, all right, okay. Then I clicked correctly. Okay.
You just have to be slow.
So here's my wild type DNA. I don't want to see it. Here's my mutant DNA. I want to see it. The way I'm seeing it is by performing single base sequencing read. Taking a sequencing primer, adding 1 base to it in an enzymatic reaction, and generating a product which I can then analyze. And what I do is I leave out the base Q, which would normally extend this primer on a C template. But I still keep the base U, which will extend. So what happens is I can't generate any product from my wild type template, but I can still generate products from my mutant template. And then when I look by an analytical method, in this case, it's mass spectrometry, when I look at the products, you can see I only see a mutant signal. There is no wild type signal. And this turns out to be incredibly sensitive. These are just experiments with synthetic mixtures. And if I put in 1% mutant sequences, you can see I get very strong peaks. 0.5% very strong peaks. 0.25% very strong peaks. But there's no background. Okay? So I'm showing you here detection of one part in a few hundred. And by perfecting this, we have detection sensitivity of close to 1 part per thousand. And if you pay attention to what I've been saying in the talk, you realize that I can't do better than 1 in 1,000. Because I only have 1,000 molecules on average, okay? So if there's one molecule that's mutant, that's cancer specific, with a sensitivity of a part per thousand, I can see it. And if I had a higher sensitivity, it wouldn't do me any good, because there wouldn't be any molecules to see. Okay, so using these new tricks, I think over the next couple of years, what you're going to see is great progress in non-invasive monitoring of cancer. And the way this is going to work, I think most people agree, is the following. If somebody actually has cancer, we do a biopsy once. You get a sample of the cancer. We look for mutations. If there are mutations that make that cancer sensitive to drugs, you treat with those drugs. You monitor the treatment by looking at DNA in the blood, which is a harmless procedure. And if the treatment is efficacious, you keep it up. And if the sequences are changing or becoming more abundant, you choose other therapy. So this is likely to change very substantially the way patients are managed. And this is truly personalized medicine. Okay? People use the term personalized medicine very loosely. But this is really personalized medicine because each person's tumor will be used to design the optimal analytical strategy and the optimal therapeutic strategy for that patient. Okay, so I'm going to stop at this point. I think it's been long enough, and I'd be delighted if you have questions. Disappointed if you don't have it. Thank you.
My name is Nadezhda. Thank you for the wonderful lecture. And could you tell at what stage of cancer detectable amounts of tumor DNA appear in the blood?
Okay, so the question, just to say it louder, is at what stage in the cancer do detectable amounts of DNA
At what stage of cancer detectable amounts of
appear in the blood?
Yeah.
We don't know.
So
We don't know. It's a great question, and it's something that needs to be tested by experimental research. And it's a very hard problem. Okay? Here's why. The right way to do that experiment is to take someone who doesn't have cancer and look at the DNA in their blood and then watch things change when they get cancer. But that requires that I'm going to know in advance who is going to get cancer. And I don't know that. So I need, what you're asking can be addressed by experiments. But those experiments turn out to be very costly because I have to start with lots of people and monitor them and then see which ones get cancer, what kind of cancer. And then I can go back and look at the blood I collected before they got the cancer. Okay. Otherwise, what I'm afraid is that there are such large variations from one person to another that if I try to do a more traditional cross-sectional study, it's going to be very messy. But there's probably some correlation between the tumor burden and the amount of DNA in the blood. But remember, in pregnancy, there's no correlation between the fetal size and the amount of DNA in the blood. So if I can draw an analogy, in addition to the size of the tumor, there probably are other variables that may even be more important. And today, I don't know what they are. So believe me, in 3 years from now, I'll have a better answer.
Thank you.
Thank you, Charles. Next question. Igor.
I'm Igor. Okay. To continue with the previous question, can we somehow improve our understanding with experiments on animals? We can insert human tumors into laboratory animals and look what amounts of DNA we can see there.
Yeah. So, so first of all, just to give a name to what you just said, there's a trick called xenografts, in which you take a bit of a human tumor and you implant it in an immune-compromised mouse, and then you can watch the tumor grow and you can try to treat the tumor, and it is a human tumor, and you can do this with a real patient. And so you can actually do experiments to help guide patient therapy by using xenografts. The thing we actually know is that the xenograft tumor that develops has the same mutations that the patient had. Okay? That we know. And that's been experimentally done very nicely. I'm worried with a mouse that I simply can't get enough plasma volume to do the analysis if things scale the way they do with the human. Now, I don't know that anybody's done the experiment, and so it's one thing to speculate. It's much more important to do the experiment. Okay? But I'm willing to guess that this is going to be tough. Okay? Too bad. I mean, I think, yeah, but if you could see it, it would be, of course, it would be great. So we have to do this different way.
Okay, my name is Sergey. I'm from Ivy Clinic and Zaragoza. And I have a question. Did you try to use liquids from cervix instead of blood to determine the short fragments of the fetal?
Okay, say it again. Instead of blood, using
liquids from cervix.
from the cervix.
Cervix.
Yeah. Okay. So the answer is yes. We have done some of that. It looks interesting, but there's a problem. So if you're going to look at liquid from the cervix, you have to be guaranteed that the woman has not had sexual intercourse for a few days, at least a few days prior to taking the sample. Otherwise, it's complete garbage.
In some periods of pregnancy, intercourse is forbidden.
Well, but those are often too late to do much clinical intervention, right? Okay. So, but it is an approach. It's an approach that looks very promising. As a matter of fact, yeah. It's just the sampling is more complex than just taking blood. But there is plenty of fetal DNA there.
What about the concentration of DNA?
Oh, it's good. It's good.
higher than in the blood.
It is, yeah.
But why do you not use oil?
The blood works really well, and is all pregnant women have blood work. So the sample change doesn't require any difference in practice. The cervical sampling is tricky because you have to. There's a lot that I can't talk about. You have to do it. It's almost an invasive procedure. You have to do it very carefully. It's almost invasive, okay? And so it requires special tools, and it's not, it's not established medical practice. It's something that we're extremely interested in, and because if I could get more DNA, then at least for those women who have relatively small amounts of DNA in the blood, this would be the alternative. I could still analyze it. It's for the future.
And you can use for differential comparison with the blood.
Of course. Yeah.
And what other lipids did you try?
What are the
What
liquids?
other biological lipids did you try?
Okay, so people have published results on urine.
Here.
Yeah. I think some of the original data was very poor. But I would not rule out looking at urine in the future. I think it's possible. The problem in urine is that the DNA fragments are much smaller than the DNA fragments in the blood. And so you need very special DNA handling methods to deal with those very small fragments. But people are making progress doing this in cancer. And so it may be worth, especially things like bladder cancer, kidney cancer, and so on, the urine is an obviously good source. So it may be worth going back and looking at this again. But again, the blood is, if you're going to talk about doing research tests, all of this is wonderful. But if you're doing hundreds of thousands of tests a year, and you're not connected to existing clinical protocols, it's very costly because you have to set up a whole system of care. So that's why I think the blood is probably here to stay. Even if the other methods eventually work well, I think the blood is probably here to stay.
We continue to trying to introduce new genetics tests in our clinic, but the problem is we don't have educated geneticists, physicians. They are the main problem of introducing new tests.
Yeah.
Can you advise us how can we solve the problem?
Right. So I think the long-term solution is to make genetics a really formal and important part of medical education, so that doctors become at least decent geneticists. And now that we know how important genetics is, and we have all these powerful tools, and I used to chair a medical school genetics department, okay, so I can tell you that doctors get a very poor education in genetics, but this has to change. The short-term solution in the U.S. and in Japan is to use genetic counseling and to more or less couple a genetic consultation to the diagnostic procedure. So, for example, in Japan, where testing started just in April, what the government decided is that prior to the test, they require a consult with a genetic counselor, and then, of course, afterwards. Okay? So that the woman, before she's tested, understands the genetics of what's being done, and then when the result comes in, it's interpreted not by the physician, but by the genetic counselor. So if you have enough genetic counselors in Russia, then that would be a good solution. At Sequino, we provide some counseling assistance to those physicians that don't have the knowledge or access to genetic counselors. But in the U.S., most large obstetric practices now at least have one genetic counselor. Yeah, but I mean, this is a real problem, there's no question. The public doesn't understand genetics, and most doctors don't understand genetics.
How does Sequinoma make this help? Right away?
How does,
How does Sequinoma help?
oh, wait, so we actually have a whole sort of staff of, I forget what we call them, medical experts that, basically talk to the physicians. So again, this becomes quite detailed in terms of U.S. medical practice, which could very well be different from things here in Russia. But with the type of tests we're doing, we are not allowed to interact with the patient. We're only allowed to interact with the physician. So we have to do everything through the physician's office. And here, it might be different.
Good. You are afraid of responsibility.
No, because this is not FDA approved, and so we're not allowed to deal with technology like that. It's illegal, probably.
My question is, in your opinion, will we can by the quality picture of the DNA in the plasma define the type of cancer? Because there are some types of cancer that we can't detect on the early stages by usual methods.
Right. So, I mean, that's a great question, and I'm going to answer it in a funny way. If I have a cancer patient, the most important thing I want to know is what drug to use, what therapy to use, whether any drug is likely to work or whether I have no choice but surgery. Okay. The way those decisions are made now are largely based on a biopsy and cytopathology. Okay, so that a pathologist knows it came from the stomach and looks under the microscope and sees certain kinds of cells. Okay. And honestly, that's a very poor way to characterize the cancer. Okay. Now, if I look in the blood, especially at an early stage, I might not even know what tissue has the cancer. But if I see EGFR mutation, I treat with tyrosine kinase inhibitors. It doesn't matter what I call the cancer. Okay? And so I think what I'm trying to say is that as I look to the future, I think increasingly cancer will be defined by the DNA sequence defect and not by the tissue origin or the way the cells look, because it really is not as important, unless you're doing surgery, not as important to know where it is or what they look like as it is to know what I can kill them with. So that's what I think will actually happen. But honestly, of course, you're right that I have to, at least in the short term, find a way to make congruent what the surgeon and the oncologist and the pathologist are used to dealing with and what the geneticist is dealing with. Okay? And that's not an easy gap to close, but that's where we are.
My name is Nicholas, I'm a student of MRPT, and my question is following. Can we say something about the previous music information about the length of DNA fragments?
Right, so you must have heard my talk yesterday, right? Yeah. Yeah. So,
but you talk about cancer.
yeah. So, in addition to everything I've told you, there is information that comes from the lengths of the fragments. The fragments from the fetus or the fragments from the cancer are short. by about 20 base pairs. Then the fragments from white blood cells and the fragments from endothelial cells in the blood vessels. We don't know why. It's shorter, but the effect's very big. It's very easily measured by a trick called paired end DNA sequencing. That gives you not just the sequence, but gives you the length of the fragment to where resolution of number of detail. So this is very useful information. And we don't understand today why this is different. The fragment lengths are such that they both appear to have come from apoptosis. But there seems to be something different about either apoptosis or post-apoptotic processing of the DNA from, coincidentally at the moment, cancer and fetus versus the host. Now, the 20 nucleotide difference is suspicious. It's 2/3 of DNA hits, okay? And it's the length of the binding site for histone H1. So the simple hypothesis, which, believe it or not, nobody has actually proven or disproven, is that for some reason, from cancer and from the fetus, histone H1 falls off during apoptosis, and so the nucleases can trim from 160 down to 140. But in endothelial cells and in white blood cells, H1 stays on. And so these things are 160 instead of 140. It's very, very curious. But just to explain to you why this is not so simple. With DNA, PCR, and sequencing, I can work with one molecule. If I have it in the tube, I can work with it. To ask whether a histone is there or not, I need a huge sample. I mean, my DNA methods are probably 10,000 times more sensitive than any protein model. So things that are just any student can do with DNA become problems that are impossibly difficult for anybody, the person. Okay? So we need to find very indirect ways of doing this, and that takes time. But we do think that this difference is very significant, and it's telling us something biologically important. We just have to discover what that is.
Can we use your method for early diagnostics for cancer?
Can we use it for early diagnostics of cancer? So I hope the answer is yes. And the issue is, how do I design the clinical trial? It really comes back to the question that was asked over there. I need to do a prospective trial because the patient-to-patient variation is so large. I'm afraid to do a cross-sectional study. I really need to do a prospective study. So I need to have a relatively high-risk cohort of patients who don't have cancer today, but will progress to cancer in the next few years. And then I start taking samples of blood. And then when they progress, for those who progress to cancer, I go back and try to see how early I can detect. It's a wonderful experiment. Somebody will pay for it eventually, but it's a very expensive trial. People don't like to do prospective trials like this because they cost so much money. Because you see, it's very hard to find that enriched population. If I have breast cancer, lifetime risk is what, 1 in 12, okay? So over a 2-year period or a 4-year period, it's 1 in 100. So to have a statistically significant 100 women progress to breast cancer, I have to have 10,000 women. And so I'm paying for enormous clinical trial. and 99% of all the patients are useless, but I don't know which one. So if you can help me figure out a way to generate an enriched population, then those trials become wonderful science and wonderful medicine. Otherwise, they just become brute force, very expensive. But somebody will do them because it's the only right way to do this. The alternative would be to try to figure out what causes the interpatient variability and then maybe control it. And people have been working on that for 10 years, and there's been absolutely no problem.
There are many other biomarkers of cancer, some proteins,
Right.
and which one is more sensitive?
Okay, so I actually, those of you who heard my lecture yesterday, I showed a few slides, but I'll just summarize it. The plasma DNA is probably 100 times more sensitive than CTCs or protein markers. There's no comparison. It's the best method. Much, much more sensitive. Not that there are more molecules, but because our experimental methods are so much better.
And about clinical trial, for example, we know that sometimes of breast cancer have huge heredity compound, and in the USA, of course, the mastectomy is rather popular, but in Russia, it is very unpopular.
Yeah.
So maybe in Russia we can use find the code for the study.
Yeah. So it's funny that you mentioned that because I was actually thinking as I answered his question that if I could take a BRCA positive population that wasn't going to really just what you're saying. If I take a BRCA positive mutation that wasn't going to undergo voluntary mastectomy, they would have a high risk of progression. But it's not that high a risk of progression. That's the problem. It's still pretty damn slow. But it's better. Yeah, it's much better.
About 80% per life. So
Yeah.
if you look for about 5 years,
Yeah.
it's rather fine, I think.
Yeah. So I mean, I think that's one possibility. The other possibility would be people who've been exposed to asbestos. So if you're exposed to asbestos, you have a tremendously high risk of lung cancer. So that's another. Yeah. But some enriched population, one way or the other, environmentally or genetically enriched, would help this a lot. But the criticism is that if you take the BRCA patients or the asbestos-exposed people, and you do a trial like that, somebody can say, it's just for that population, it's not general. And so, there are trade-offs here. I hope at some point we can find somebody to support a very expensive longitudinal study and do this right.
Thanks.
Okay, I think
Is there a correlation between other diseases and high DNA fermentation rate?
it's not really known. There are only 2 other cases that I know that have actually been experimental studied right now. And that's, let's see if I can think of what they are. Yeah, so people who've had bone marrow transplant, or people who've had other organ transplants, you can then use the sequence differences between the donor and the recipient to distinguish between DNA fragments that come from the donor and the recipient. Okay? And so there is data on transplant rejection, and there's data on how a patient progresses after a bone marrow transplant. Those are the only 2 other cases that I know right now. And honestly, I certainly am sure that in transplant rejection, the amount of DNA in the blood goes up tremendously. But of course, that's massive cell. Oh, and the other example, which is not a disease, is trauma. Yeah, of course, you travel, there's lots of information.
So here we would like to conclude our online discussion, and we can probably take the discussion offline. So please, those of you who attended our online translation can send their questions, can post their questions online, and we'll relay them to Dr. Kantort. Thank you very much, and have a good day.